second part for that chapter 24, we're going to look at why the lipids and how they are used in the body's metabolic pathway. You have three different sources. These all come from the digestive tract as food is broken down. They also result from adipose tissue where excess lipids that were coming from the diet have been stored. And they also come from the liver where lipids can be synthesized. Whatever is their source, these lipids must eventually be transported in the blood to reach the cells and help for the production of energy. So looking at here, things you should memorize here, the lipids in the diet, fatty acid from storage in adipose tissue, the triacylglyceride synthesized in the liver, cholesterol also synthesized in the liver, and the cholesterol that comes from dead cells. You see at the bottom of that slide, this is the bloodstream, and that's the way they can be transported. So the first one on the left are chylomicrons. Then you have a transport by serum albumin for fatty acid from the storage that you find in the fatty tissue or adipose tissue. And then the VLDL, LDL or HDL, this has to do with the density. So VLDL, very low density lipoprotein, LDLs, low density lipoproteins, and HDL, high density lipoproteins. You need to know those definitions. So, well, what happened? Fatty acids that are coming from adipose tissue, or the fat in general, like we call it, associate with albumin that you find in blood plasma, and it can actually bind up to 10 fatty acid molecules per protein molecule. Everything else is carried by lipoproteins. And remember that lipids are less dense than protein, so density of lipoprotein depends on the ratio of lipids to protein. That's why you have VLDL, LDL, HDL. So we have 5 major types that I would like you to memorize. The chylomicron actually are the lowest density of all. Less than 0.95 gram per cubic centimeter. I'm not going to ask you to memorize the density exactly, but I need you to know the difference between the 5 major types. The next one are the very low density lipoprotein. They carry triacylglycerol from the liver, where they were synthesized, to tissue for storage or energy generation. Intermediate density lipoprotein, IDLs, which you rarely hear about, carry remnant of those VLDLs from peripheral tissue back to the liver for use in synthesis. And then the one that you probably will remember from cholesterol and a ratio that will be taking apart. The amount of cholesterol is one thing, but also the ratio of cholesterol to LDL or HDL. So which one is the good cholesterol, enter comma, which one is the bad one? The good one is the HDL because it transports cholesterol from dead or dying cells back to the liver. So that means that you have less cholesterol in the bloodstream, and that prevents the risk of atherosclerosis. Low density lipoprotein transport cholesterol from the liver to the tissue, so that will increase the amount of cholesterol in the bloodstream, and you can have all those cardiovascular diseases such as atherosclerosis, stroke, or heart attack. So we do need triacylglycerol. They are very important for the cells membrane, but also long-term energy storage, insulation of our bodies, and cushioning of our internal organs. So when you look at the overview of the metabolism, I would like you to pay attention to that diagram and read it carefully. This is just a summary. I cannot go with everything in the narrated lecture. But here, look at that diagram on the right side. Triacylglycerol can be converted into fatty acid, and those can enter the acetyl coenzyme A citric acid cycle. This is to make energy. So that hydrolysis, the one you have here on the far left, occurs when chylomicron encounter lipoprotein lipase anchored in capillary walls. They are going to be used most likely to make energy, but if you have enough energy, well, those are going to be converted back into triacylglycerol for storage. Where does it take place? In the adipose tissue. But then when the cells need energy, those fatty acid carbon atoms are activated and then oxidize as acetyl coenzyme A to enter the making of energy. Look at here where acetyl coenzyme A can either generate energy, and that's from the citric acid cycle, oxidative phosphorylation, but also can start as the starting material for lipogenesis, ketogenesis, and the synthesis of cholesterol, from which all other steroids are made. So triglycerol from adipocyte. Adipocyte, they are the cells that contain that adipose tissue, so they are the ones that store that energy source. When stored triglycerol are needed as energy source, lipase, that enzyme within the fat cell, are activated by the hormone level variation, low insulin and high glucagon. Those stored triglycerols are then hydrolyzed into fatty acid, and those free fatty acid and glycerol can be released into the bloodstream. So what's the fate of those dietary triglycerols? Well, they can undergo hydrolysis to fatty acid and glycerol, resynthesis of triglycerol for storage when there is a lot of energy, or conversion to acetyl coenzyme A when energy is needed. Now, those are some of the steps. So remember, we have glycolysis, which is to make energy, gluconeogenesis, making glucose from non-carbohydrate bases, and triacylglycerol synthesis, which is also energy storage. Note that acetyl coenzyme A can actively participate in making triacylglycerol, making ketone bodies, so ketone body synthesis is called ketogenesis. Genesis is to make. Synthesis of sterol and other lipids that are necessary, or going under the citric acid cycle and oxidative phosphorylation for the generation of energy. Now, the passage of fatty acid in and out of storage in adipose tissue is a continuous process, and that is what maintains what we call homeostasis. So the mobilization of those triacylglycerol, well, if you think about it, when you are digesting a meal, blood glucose level return to normal, insulin levels drop, and glucagon levels rises. Remember, insulin will lower the amount of sugar in the blood. Glucagon will do the reverse. They are antagonistic hormones. The lower insulin level and higher glucagon level activate triacylglycerol lipase, that enzyme that controls hydrolysis of the stored triacylglycerol. And then if you look at this, when those glycerol 3-phosphate is in short supply, fatty acid and glycerol produced by hydrolysis of stored triacylglycerol are released to the bloodstream for transport to the cells where they can start the making of energy. Otherwise, those fatty acid and glycerol are going to cycle back into new triacylglyceride for storage. Now, this is a very good summary. Look at the food. We started with carbohydrates, now we look at the lipids, and you see next step will be the proteins. Everything can help for the making of energy, and if we have excess of energy, they are going to be stored just in case we need energy later.